So I will be talking about offline-rTMS techniques and how they can be used to gain insights about functions of brain regions. So this will be based on my very recent rTMS project, and I will explain what these terms mean as we go along in the presentation. So first, I wanted to motivate the reasons behind why we chose to do TMS at all. So I'm interested in characterizing the brain networks which are involved in inhibitory control. So inhibitory control or response inhibition is the stopping of prepotent responses in order to select a behavior which is more consistent with achieving the current set of goals. So, and we are interested in both how people stop their actions, like motor actions, and also how people stop their memories or stop memory retrieval in the context of adaptive forgetting. So we have some evidence in the lab from fMRI, from fMRI studies. For instance, there was this meta-analysis which looked at previous fMRI studies, and it showed that there are similar cortical regions which are involved when people are engaged in either stopping the actions or when they're engaged in stopping memory retrieval. And these are different kinds of motor and memory inhibition tasks that we use in the lab. And recently, we've also shown that in the lab, this being published work, it shows that using dynamic causal modeling, you can show that certain prefrontal regions here are involved dynamically in downregulating their respective targets, depending on the task that the participants are performing. So now the natural next step or next question that I wanted to ask was, can we actually provide more causal evidence as to the involvement of these prefrontal regions? For instance, the right dorsolateral prefrontal cortex, can we modulate activity in that region and see how that affects people's ability to stop their actions or stop the memory retrieval process? So which is why we decided to look at non-invasive brain stimulation techniques and specifically at TMS. So yeah, we all, I think, understand to some extent how TMS works in terms of what we're doing. So it involves a coil that we place on the scalp, and the coil is basically a current carrying conductor. The direction of current in the coil changes very rapidly, meaning the magnetic field associated with the coil changes, which means that there are induction of secondary currents in the underlying brain region. So which translates to change in membrane potentials or potential across membranes, basically. So TMS has been shown to be quite focal in terms of what regions you're targeting. And there are two classifications of TMS. One is online TMS methods or repetitive TMS methods, wherein the TMS is administered when people are engaged in doing the task. And offline TMS methods are usually when TMS is administered before the task is done. So you administer the TMS technique first, and then participants do the task. And usually the effect of the offline TMS lasts for some time, and people perform the tasks in that time window. So what are the specific advantages and disadvantages of the two methods? So if one is interested only in, or if the primary question is to see if a particular brain region is involved causally for creating certain behaviors, then offline-rTMS is an easier way because then you can just modulate activity in that brain region and see how that affects the task performance downstream. But because in online rTMS, you could have effects of the TMS itself, which can kind of change task performance during the task. So during each trial, there's the effect of pulses being given, which can be a source of distraction, for instance. But if you're interested in the how certain regions come together and create certain behaviors, then, or if you're interested in the mechanistic modeling itself, then online rTMS methods might be the way to go. So I will be focusing on offline rTMS methods. So one popular set of protocols of offline rTMS are the theta burst protocols. These were first introduced in 2005. So essentially, it involves trains of 3 TMS pulses. So each train consists of 3 pulses, and the inter-train frequency is 50 Hertz, and the inter-train frequency is 5 Hertz. Now, there are 2 major kinds of theta burst protocols. One is intermittent theta burst, wherein these trains are administered for 2 seconds, and then 10 seconds later, the next set of trains are administered, and so on and so forth. So the total number of pulses administered in this way is 190. And then we have the continuous theta burst protocol, wherein it's just continuously administered theta bursts. So all the trains are administered continuously for 40 seconds. And so that leads to a total of 600 pulses being given. So it was initially shown using motor evoked potentials recorded from the motor cortex that the ITMS, the intermittent TMS, intermittent TBS protocol produces facilitation in the target region for about 20 minutes after the administration of the TMS, while CTBS produces an inhibitory effect or downregulation in the region targeted, and this lasts for about 60 minutes. So this was originally shown in the Huang paper, and it has then been subsequently taken up, and that has been shown in many other studies as well. However, there's a lot of variability in terms of the results, and also even in the same set of studies. So TBS variability can be both intra-subject and inter-subject. So however, the intra-subject variability is quite small compared to inter-subject variability. So the intra-subject variability is usually down to circadian influences and cortisol responses, for instance. The inter-subject variability, though, can be due to a variety of factors, so including like age and cortical network activity, and like inherent variability of neurotransmitters in the brain, and so on and so forth. So what I thought I would do was to kind of list out some good practices. These are things that I had to think about when I was designing my CTBS study. So so this might kind of maybe lead to less variability in terms of results, or at least more comparability of the different study designs. So one thing that people need to think about would be the study design itself. So as we saw, intra-subject variability is lower compared to intra-subject variability is lower compared to inter-subject. So it would be better if you had like within subject designs rather than group designs. And also the number of participants being tested and power is something that many TMS protocols need to think about, I think, because a lot of protocols historically have been, a lot of studies historically have been underpowered. And also it would be nice to look at like double-blind studies wherein the researcher who's administering the experimental like instructions is blinded or is not aware of which group the participant belongs to, because that could also have effects on how the experiment is being administered. And using individual structural scans for neuronavigation is definitely a must. And what would be even better is to use functional localizers for the particular like tasks that you're interested in. And using those localizers for neuronavigation would definitely produce like better results. And there's also emerging evidence which suggests that like the people who respond to these kinds of protocols in the way that we expect them to, the responders, they might have a set of like, there could be ways to identify them beforehand. So one is by using their resting state parameters. So that is something that people could look into before designing TBS experiments. And again, at what intensity should the stimulation be administered at? So I think more or less now people accept that 80% of the resting motor threshold is a good intensity value. And again, you can have different ways of measuring the resting motor threshold. One is via just visual inspection, which is what we saw in Marius's video as well, where we saw the thumb movement. But then you can also measure it by using like electrodes. You look at the motor evoked potentials. And if one decides to do that, then that could also be used as a proxy for elucidating responders. So we could have sessions where we perform CTBS in the motor cortex and look at the motor evoked potentials, if there's a reduction in that or not. Again, the assumption there will be that people who are responders in the motor cortex will also respond similarly in other areas of the cortex, which needn't be true, but it would be interesting to see if there's a correlation between how these different like cortical regions respond. And finally, there's also some evidence to suggest that MEVs could potentially, again, be used as biomarkers for identifying responders. So for instance, CTBS has been shown to very selectively act on the I-late waveform of the MEP. So they're like B waves and I waves and I1 waves. So like different TMS protocols or like could differentially act on these waves. And depending on how selectively people produce these waves in the first instance, could also suggest or give us some indication as to if they will be a responder or not. And coil orientation is something very important. And another thing which people kind of forget is that it also depends on the current direction, and different coils actually have different current directions in the sense that it could be clockwise or counterclockwise, which I didn't first know when I started designing the study. And also there's a lot of work now which suggests that even if we are targeting the same brain region, the coil orientation can affect the networks that you're hitting. So TMS could actually differentially act on different networks that this region is part of based on the orientation of the coil. And another thing which everybody could do is investigate or visualize how the electric fields actually were created after CTBS by either having readouts like fMRI or EEG readouts. And again, this is one advantage of offline TMS methods, because you can perform the TMS outside the scanner and then immediately take people into the scanner. So that's relatively easier than having simultaneous TMS fMRI experiments, for instance. And you can also do some post hoc like modeling after the TMS is performed. So if we have the structural scans of all the participants, and if we know the coil orientation and the region of stimulation, then we can use SimNIBS, which is a simulation software for simulating how the electric fields would have been for that particular participant. And then we can kind of look at which kind of networks then we actually ended up hitting using the CTBS protocol for that particular participant. And another thing that people need to think about are controls. So this was, again, a few years before, sham coils weren't very effective in being sham coils, but now I think we've made progress in that area, and now there are better sham coils available. And so, for instance, in the CBU, we acquired this new sham coil, and we've kind of run pilots where we asked people to judge if they thought it was sham or not, like actual stimulation coil, and people couldn't really tell. So the painfulness of stimulation, the noise and everything was similar. So that is encouraging. And another thing is control regions. So this is something, not all studies have control regions, but if you do have that, then what kind of region would you use as a control? This is something that we need to think about, because initially, I think many people in the field thought that using the homologous lateral region would be a good control. But then for CTBS, for instance, that wouldn't work at all, because now there's a lot of evidence to suggest that CTBS on one side of the brain might actually create facilitation effects on the homologous opposite lateral side. So, and more now, many people seem to think that vertex would be a good control region, but we'll come back to this point again at the end of the presentation. And finally, it would also be good to collect some subjective measures of stress or pain that participants experienced during stimulation for each group, because if you're primarily looking at only behavioral readouts, then differential amounts of stress or pain can also kind of affect those readouts. And so you might just be looking at the effect of stress rather than stimulation itself. So I will now briefly come back to my original motivation. And so we did end up doing this study in the CBU during the pandemic. So that is the setup. So actually for half of the study, we used this room because we had to run studies during the pandemic. So we needed a large room. And then we shifted back to the TMS room. So those are some snaps from the TMS room and the CBU now. And so we did think a lot about the good practices that I listed about, but we couldn't follow all of them. For instance, we don't have a functional localizer. So we ended up using MNI coordinates from the meta-analysis. So we used this set of coordinates for the DLBFC. And we did have a double-blind design though, which was good. And we had 2 sessions counterbalanced on different days. So in each session, participants either did a motor inhibition task, which was a stop signal task, or a memory inhibition task, which was a think, no think task. And so in each of the 2 days, the training on the task was completed before CTBS was administered. And then the main task that we later looked at was, so people did the main task in the 60-minute time window after they were administered with CTBS. And again, we couldn't use a within subject design because of various task design constraints. So we had to go for a group design. So we had 3 groups. So one is the RDLPFC group, which received stimulation, which received CTBS in the correct spot using the stimulation coil. And then we had a vertex group, which we used as a control region. So they received this CTBS at the vertex though. And sham, which received stimulation at the correct spot in the DLPFC, but we used the sham coil instead of the actual stimulation coil. So initially our plan was to have 30 participants per each group, but then we ended up with 24 because of the pandemic. So we just decided we'd go with that. So I will share some preliminary results. So these are very preliminary, and also I'm not going to go into detail into other parameters that we were looking at. So I just thought I'll share these because it's interesting for the discussion as well. So we looked at forgetting after the memory inhibition task amongst the 3 groups. And so the more positive this number is, it means more forgetting happened. It means people were successful in inhibiting the memories that we told them to inhibit. So we see that both the RDLPFC group and the vertex group show diminished forgetting in the memory inhibition task, while sham group perform as expected, as in a normal TMS, a normal TNT task. And this led us thinking as to why the vertex is also behaving similarly to the DLPFC. So is it just because the active stimulation is creating these effects, or is there anything else going on? So one thing that I was thinking about before the experiment as well was that the vertex stimulation could potentially affect readouts in the ACC, for instance. And the ACC is a region that we think is responsible for in the inhibitory control network as well, and it's responsible for looking at intrusions. So intrusions is when these memories come into awareness, even though people are trying not to think about them and trying to stop the retrieval process. So in order to see if there are differences in how the groups behave with respect to the intrusions, we also collected like trial by trial intrusion score from every participant. So we first looked at all the trials together, and we didn't particularly see any difference between the 3 groups in terms of their proportionalized intrusion scores. But then in this particular experiment, we have 2 sets of stimuli. So one set of stimuli are scenes of the memories which were encoded were negative, while the other set was neutral. So we then looked at those 2 separately. So if we just look at neutral, again, there wasn't much of a difference across the runs in terms of the reported intrusion scores. But if we looked at negative, then there was a difference, and it was very interesting. So we see that in block number 2, both vertex and DLPFC group report higher intrusions than sham. And for the vertex group, this remains high even at the end, and it's actually significantly different from the sham group. So this has led us thinking into if the vertex simulation did not just not do anything, but if it also created changes in the functional connectivity of regions that we're interested in, which could have led to these results. So emerging evidence from other studies, which suggests that vertex stimulation might change resting state connectivity between ACC and DLPFC. So this is something that we need to look into in more detail, I think. So for example, resting state functional connectivity studies and also SIMNIFS modeling can ascertain how CTBS on vertex could lead to changes in activity in other regions of the cortex. And it could also depend on the regions that we're looking at and the type of long range connections between the vertex and those regions. And this could also, food for thought as to if vertex could be used as a control region for any TMS as well. So that's something that I want to look into further, and I'm happy to have thoughts on that. And also we are currently analyzing the other results from the study to see if this hypothesis actually holds up in terms of if the vertex simulation is only creating these kind of modulations because of the intuition scores, or if there's anything else happening as well. So I would like to thank Meera. She was part of the data collection and the participant recruitment. So yeah, and very, very helpful during the entire experiment. So, and also Mike and my funding agencies, and the CBU technical staff, the reception, Brainstorm team, and MR team. They've been extremely, extremely helpful for helping us run through the pandemic, which wasn't an easy task. And also my wonderful lab. Thank you. And any questions?